Again, hello and welcome to all of you that are joining this program dedicated to metagenomics. Now, before we start talking about metagenomics itself, I want to mention a little bit about the OmicsLogic programs. The OmicsLogic programs are designed to introduce non-bioinformaticians and people that are not coming from an engineering necessarily background to the complexities and the opportunities with omics data in biomedical research, as well as all life sciences. We believe that today biology is moving forward with a lot more data being generated than we have the capacity to analyze. So it's a priority for us to enable the future generation of biologists with tools that they can use to solve this challenge of big data. So we're excited to set up a series of programs that are focused on different types of data. And this program specifically is going to be focused on metagenomics. Now, what is metagenomics? Meta essentially is a prefix that stands for collection of different elements that comprise a single entity. So a lot of times you might hear words like meta-analysis, which means analyzing data from multiple sources or analyzing perhaps something that was published in a variety of different publications. Metagenomics really talks about a collection of genomes, a collection of genomics is of course talking about genomic data, so a collection of genomic data about different organisms. And a lot of times when people speak about metagenomics, we're really talking about microorganisms. Now, microorganisms are much more abundant in the world than macroorganisms like humans and other species. And so the field of metagenomics is a field that has been rapidly evolving ever since the microbes were first discovered when we had the microscope, when the microscope was invented. And today we have a new level of detail that is possible due to availability of genomic data. So this program is a 2-month program that will start in November and will last all the way till the end of December, giving all of the participants the opportunity to understand the different challenges and opportunities in metagenomics, as well as to have hands-on experience working with data, metagenomic data, that you will learn how to analyze, how to interpret biologically the meaning of the analysis, and then also to demonstrate your capability by designing a project of your own. Now, we're not expecting everybody to necessarily come to develop a project of their own, but we are excited about the types of projects that participants of previous programs have been able to develop as they participated in the program. So this OmicsLogic program is starting today, and this is one of the free sessions, and we are doing this session to encourage people to understand what the program is all about, how it will help them wherever they are in their journey, and what are some of the topics that we will cover. So we'll really explore the role microbes play in human well-being, health, and disease. And we have really worked hard at designing this program to be available fully online and to be rewarding to students and all the way to experienced researchers that can utilize the types of data and the analysis methods to get biological insight out of this high quality, high throughput data. We plan to cover 2 major topics in metagenomics. One of them is the analysis of 16S and 18S ribosomal RNA that can allow us to study the hypervariable regions and understand composition of the microbial community when we talk about 16S RNA. But also we'll talk about other types of microbial genomes, and we'll talk about genomics in the context of and understanding how these different strains of viruses and bacteria interact with environmental challenges and develop, and how can we study that using genomic data. So we are starting today with an overview of the program. So what we really want to talk about are two things. We want to talk about expectations. What might you expect from participating in this program? And talk a little bit more about the schedule and deadlines and important dates of this program. So what we will do is we will meet online, just like today, to cover a variety of topics. And typically we will go from more theoretical topics to then more and more practical, where we will learn how to analyze data, and we will have a chance to review specific data sets. These data sets are all going to be from the public domain, so everybody will be able to read the publication about how the data was generated, understand why the data was generated, analyze the data, and then compare the results to what the authors of the publication showed in their methods section. So really the program is going to cover a variety of theoretical background information with some practical skills, practical ways to analyze data, and then also conceptually how to think about different types of projects that are possible with this kind of data. So we will talk about, like I mentioned before, online sessions, practical, and they are going to be supplemented by online materials that you will have access to on our educational platform or portal, where you can find a variety of different projects and a variety of different online lessons and really learn on your own schedule. Now, some of the topics that we will go through include understanding the microbiome, how microbial communities live and function together, and how they affect agriculture, organ function, and health. So we'll talk about the microbial community and how it functions. We'll talk about sequencing, including 16S and 18S ribosomal RNA sequencing, but also genomic sequencing, next generation sequencing in general, and how the development of these methods are really allowing us to understand better the microbial communities, and what does this understanding actually produce? How can we apply this understanding of the way the microbes live and work together between themselves and with the host? How can we apply and use that information? Then we'll talk about several ways to analyze metagenomic data. So there are two main aspects of analysis of metagenomic data, where we will talk about the challenge of accurate assembly of a metagenome, a genome of a specific, for example, variation, a species. For example, as you know, in viruses, there's this concept of quasi-species, really a diverse population of the same species, but that has a lot of variance, and so therefore they are not represented by single genomes like we typically think of human as the human genome. Well, if you talk about Ebola or you talk about flu, you're really talking about a community of genomes. And we can still study this community of genomes by looking at the variation at the genomic level, understanding that there's a representative of a population that we are looking at. And we will talk about the non-genome-based metagenomics, so really metagenomics as analysis of the microbial community based on their variability. So we'll look at genome composition in a sample, understanding that this is a sample coming from a whole variety of species that work and live together and function together. And so we are looking at their function as an entity, as a microbiome. And so we'll then understand the microbiome and how it really functions, and how we can study variation in composition as well as variation at the genomic level to understand infectiousness or different phenotypes related to infectious diseases. We'll talk about phenotypes that are related to, for example, how much fruit a certain plant produces based on rhizobacteria, how infectious and how fast the disease spreads based on genomic understanding of the genome variation, as well as how can we link the composition of the microbiome, for example, with a stress response or some kind of a well-being or a healthy versus sick condition, as well as how do we understand resistance to different drugs, including resistance to antibiotics. So those are some of the topics that we will cover. And for each one of these topics, we will provide you with an example, a data set that could be studied in the context of that topic. So for example, we will have several projects. Each project is taken from a publication. So we'll have access to the protocol of how they generated the data, how they sequenced the data, how they analyzed the data, what are the conclusions from that analysis, and we can repeat the same steps to really understand what we can learn and how we can leverage that example to think of similar types of studies. One big aspect of metagenomics and the popularity of metagenomics is the emergence of 16S metagenomic sequencing. So amplified 16S ribosomal RNA sequencing has really reduced significantly the cost of sequencing and therefore study of a microbiome. So in November, we will start by talking about the aspects of sequencing. How does this sequencing really work? So we will talk about how this could be a very low or moderately costly study. It really focuses on a 1500 base region that could be extracted and then amplified to sequence either the full, typically the full sequence across the whole sample. And you can therefore study this hypervariable region that can provide species-specific signature that are very useful for bacterial identification. So those are some of the methods of sequencing where you can look at amplicon of just specific regions of 16S ribosomal RNA. You can study the whole length of 16S ribosomal RNA, or you can also even do the full genomic kind of a standard sequencing without amplifying anything. And we'll look at different examples of how these different types of sequencing methods can be used in the context of these studies. When it comes to sequencing the hypervariable region, we're really talking about a portion of the ribosome. So we're talking about the RNA that encodes the bottom part of the ribosome when it sits on the RNA. And we will look at how these different regions, so you can see those regions over here on this side, this is for E. coli, you can look how these regions could be used to very specifically understand the composition of the microbiome. They are highly conserved within the species, and they're pretty short, and so therefore you can sequence a lot of them so you can have good coverage, and at the same time they are very fairly well annotated. So instead of trying to assemble whole genomes of different representatives of species within your sample, you're able to just use a database and then learn about the composition and the different proportions of species, phyla, and family within your sample. So that's when we will talk about the microbiome as a functional bacterial community. So really the microbiome is a composition of fungi, bacteria, and viruses. It's essentially all the microorganisms in a single sample that it has stable condition within a specific environment. So this could be an ecological sample, so sample from water or dirt. It could be a sample from the gut, for example, or it could be a fecal sample. So we think of the microbiome as a functional community. It has a specific set of functions, and it is really defined by the microbiome stable condition. And so we will also look at how the stable condition can change as a result of different environmental factors. For example, we can think of the food, the intake of nutrients, the food, processing of that food inside the human gut as being a process that is fairly stable, and it could be disrupted by the change in diet. So one of the projects that we will look at is actually how does the changing of diet, even when it is not long-term exposure to really change the physiology of the host, but a short-term a kind of transplantation of a microbiome can change behavior. And so this is a very interesting area that really points to the functional aspect of the microbiome. The microbiome can really, in the gut, the microbiome in the gut can affect the way that we function, we think, we interact. And that's a fairly new area that is being studied now in terms of the microbiome. Now, the microbiome actually has these stable communities in a variety areas around our body, and we can study them as these stable communities. So skin microbiome, vaginal microbiome, gut microbiome, different types of microbiomes. And so now you can think of those as ways to study how different processes occur that are very critical to human health, right? We talk about, for example, skin diseases, and we talk about the oral microbiome and aspects of oral cancers or dentistry and things like that. We can talk about the gut microbiome and how it relates to intake of food, ability to process specific types of food, and of course, how that affects our response to different treatments, because of course, a lot of the treatments come through the gut. So we take pills and then through the stomach that get into the bloodstream, right? So a lot of that has to do with that functional microbial community. So we can use a database that essentially tells us how taxonomic classification of these different microorganisms can essentially work together. You can see here composition of different types of bacteria, how the composition could be studied to understand the health and the processes, the functionality of these, so to say, organs, the microbiomes associated with different parts of the body. And so we will look at different examples that will help us understand how this actually works. Now, there are some challenges associated with analysis of such data. A lot of these challenges are related to the understanding of how sequencing actually works. And so sequencing, when we sequence these hypervariable regions, we will have a challenge of aligning the short reads and then making sure that, so typically this is paired end reads, so we have more reliability of the sequence itself, but nevertheless there are a number of methods that have been designed to address some of these challenges. How to identify properly reads that were aligned correctly, how to identify the hypervariable regions correctly, how to map them to the database, which database to use, how do we create these organizational taxonomic units or OTUs, and how do we really then use the composition based on these OTUs using clustering techniques, etc. So those are challenges related to the 16S ribosomal RNA, but also we'll talk about other challenges. If you take a sample and you sequence total RNA, what are some of the challenges there with assembly, with K-mers, so really assembly of genome entities that are not necessarily mentioned in databases. So we'll talk about some of those challenges as well. And it's going to be very important for us to really understand, again, the theoretical aspects of what these challenges are, but then also to look at practical examples. How do we use, in a practical sense, this or another type of data, and how do we go from sequencing the data, mapping the data on some database or on a collection of genomes, and then actually getting some biologically meaningful information that is significant enough for us to say something about something like the health of the gut or some of these other examples that we talked about. So as I said, there is a variety of different databases that are available to do so. So we have different databases like Green Genes, Silva, NCBI. NCBI, of course, hosts a lot of data that has been submitted on a variety of different genomes. But just taking all of the genomes and mapping everything that we have in our reads on those genomes, there's going to be a lot of overlap. So the data is going to be noisy. So some of these databases were actually more curated and validated biologically to help us reduce the amount of noise that we have in our data. And we'll look at some of those examples where we will compare the different databases and really try to run our analysis so that we understand in which case, which database, which reference database is going to be more useful. So we'll have a whole session dedicated to kind of a review of some of these databases. And we will particularly look at how to compare the results. So Silva, for example, is a comprehensive online resource for quality checked and aligned ribosomal RNA sequence data, which is important because it really validates a lot of these so biologically actually uses the sequences that are in the database to make sure that biologically this works. And this is an important aspect because we will look at how a lot of noisy data coming from the availability of this genomic sequencing can actually be more confusing because of chimeras and because of different wrong assembly of organizational taxonomic units. Now, on the other hand, we will also look at examples of specific projects developed by NCBI and FDA and other regulatory agencies that actually are more focused on detection, presence, or absence of specific species that that have been understood as being harmful to a specific process. So an important aspect of that is really taking and looking at the database design from the standpoint of how it is going to be used. So we'll take a look at some of those examples. Now, a way to apply practically this type of understanding, this type of knowledge about how the sequencing works and the types of data that you can generate is to actually try and run a pipeline. So the data2 is a good example of a pipeline that is specifically designed to address a lot of the challenges that we talked about up until now related to 16S ribosomal amplicon sequencing. So we'll take a look at that pipeline and we will apply it to a number of different projects to understand how it addresses some of these challenges that we discussed. And I want to mention how the program is built around, like I mentioned, theoretical concepts, project examples, and some practical tools. Now, a lot of times you will find that courses require all kinds of installation of software and really a variety of different packages, and that's where a lot of programs fail like this. So we would like to believe that our program has addressed that challenge very efficiently by putting all of the packages that are required behind a visual interface that is easy to use. Now, this is a functional, usable interface that you can apply to Exemplar projects that we will go through. You can apply to public domain projects that you will source from places like NCBI. And you can also use it for your own data. It's a fully developed research platform. So I want to talk a little bit about that platform so that we can understand how it works. So the program itself is going to consist of online sessions like this one It will also give you access to online materials that Mohit pasted links in the chat to some of the courses that we have online for your independent learning. And then another aspect will be actually going through data sets and using those data sets in the context of analysis. So I just wanted to make sure that it's fairly straightforward for all of you to see how this works. So let's take a look at this platform really quickly in the context of metagenomic analysis. So you can see that the server T-BioInfo platform is available for anyone. You can actually request access for research use or for educational use just by filling out this form right here. But you can also learn about, in the context of this program, about a different, several different areas on the platform are going to be related. So for example, here you can see the amplicon 16S and 18S ribosomal RNA, and we'll take a look now at just one of the examples. Here you see the full sequence assembly of total RNA and use of that data by mapping it on a variety of different genomes. And you also have a number of sections here related to virology. So we will actually cover several of these sections in the program, and we will help you also find the packages that you might need if you would like to set up your own pipeline, design your own pipeline, or run this pipeline on your own server. We will also help you identify packages in R and design the pipeline to work on your end if you're interested. But in the context of this program, we'll primarily rely on this platform to understand how to process the data. So let's just take a look really quickly at a demo. Now, anyone can request a demo for free, and you can look at how this works really to understand the concept behind the logic of this pipeline. So if we click on Start, here we can read a little bit about what we've been talking about, and here you can see what the amplicon sequencing of 16S hypervariable region. And you can understand a little bit about these conserved regions. Really the types of pipelines are really designed to address some of the typical challenges with this data. So once we click on OK, we can start the data-to pipeline. So the data-to pipeline starts with filtering. So you really have to reduce the number of reads to reduce the noise. And you can read here in each one of the pop-ups, you can actually read a fairly detailed description of what the pipeline does with explanations. For example, this is an explanation of read quality, where certain reads are high quality and they're kept. Some reads are low quality and they are not kept. And then you can see also the output of this step would be kind of an assessment of general properties of reads for this kind of filtering. And you can also see a full description and a detailed tutorial on how to implement. So for example, if we go here, you can see how to install this package yourself. This is something that is based on a number of packages in R, so anyone can actually install this pipeline. But let's click on OK. And now we will look at several other steps that help us prepare the organizational taxonomic units, OTUs. So now we will go through consensus of OTUs, and really data-to pipeline was designed to improve quality of 16S metagenomic sequencing. So you can see, for example, the core data-to method corrects substitution and indel errors, but chimeras remain. Fortunately, the accuracy of the sequence variants after denoising makes identifying chimeras simpler. And so really, it is all about having high quality data. And then once we have high quality input data, we can accurately map that data to a reference database. So here you can see that this was done by mapping the data on the Silva database. And you can see that really what the Silva database contains are just sequences of these hypervariable regions for a variety of different species. Click on OK. Now we need to construct the phylogenetic tree. So phyloseq is an R package that allows us to construct this tree. And what we will see are the actual relationships between the abundance and the type of level of organizational taxonomic units that we have chosen, species, phylum, family. And then we can also think of a comparison between our samples. For example, we can think of the composition inside a sample, which is called alpha diversity, or the comparison of two samples or two groups of samples that could be called beta diversity. So what would be the output? The output of such a pipeline really will produce a table of abundance where we will see OTUs, these organizational taxonomic units across our samples. We will be able to identify what those OTUs represent in the organizational taxonomic units. And then we will also have the ability to see composition, study the composition of the microbiome. So now we can click on OK. And here we have some outputs. And those are the outputs that we will be studying. So now the question is, what is the data? What are these samples? How do we interpret what we have been studying in the context of the project? So that is a little bit about the practical use of the platform. Again, the platform itself is really comprehensive and allows us to process these types of specific data sets that we looked at here. It also provides access to some machine learning and data mining tools, has some very useful utilities for annotation and visualization of data, and also has a lot of different types of data that you can work with. So really the platform is designed for multi-omics data. And if you think of the microbiome, or if you think about any microbial data, we typically think of that microbial data in correlation with some other type of data. And one way to study this is really the host microbiome interaction. How does the change in composition of the microbiome affect the response from the host? For example, the immune response from the host, or the response in terms of, for example, response to treatment, right? Development of resistance. How does it affect the population scale when we look at, for example, spread of an infectious disease in a geographic endemic? How do we look at that region? How do we look at speed of progression of that disease? How do we look at response from different types of populations, etc.? So really, it's all about the relationship between the host and the agent. So going back to our overview here, as we looked at, we will study the data to pipeline and really understand what this data to pipeline does. How do we merge it with other packages that are going to be useful for visualization, downstream analysis, validation? How do we incorporate the T-Bio platform provided interface with other types of tools in R, in Excel, in other environments where we can really analyze the data further to get granularity from our data to be able to interpret the meaning of that biologically. So in the sense of understanding what this ribosomal RNA data can give us, we really can look at several different examples, understand the outputs, understand the relationship between the hypervariable regions, and this type of study that looks at composition or alpha and beta diversity. So then after we've kind of gone through the basics, right? We talked about different types of microbial communities. We talked about different types of organisms, viruses, bacteria, fungi. And then we talked about how we can study different types of sequencing techniques that allow us to study different aspects of these microbial populations. We will start thinking about projects. And so what we've done is we've prepared a number of exemplar projects The American Gut Microbiome Project, called just simply the American Gut, is a good example. This is a citizen science project that was designed to collect samples from participants like you and I. We can actually all do it at the cost of $99. Any one of us can participate and contribute our samples to this project. Really what they did is they studied the microbiomes from people from all over the US. And I think now they've expanded to other countries as well. We can have access to that data, and people also answered questionnaires. They answered questionnaires about what they eat, what is their ethnic background, what is their different psychological traits. So we can start looking at the type of data that we have available and how it can help explain some of these self-reported phenotypes. We also will look at several other examples that we've prepared as fully digested projects for specifically this program. So we'll look at gut-brain axis, again, the relationship between microbial composition and behavior as studied in animal models in mice. We'll look at Ebola, and we'll look at tuberculosis. So when we talk about tuberculosis, we really have a project, a collection of a lot of samples. There have been recently a number of studies on microbial resistance to antibiotics, and tuberculosis is a good example. It's a mycobacterium, and it is treated with different types of treatments in different locations geographically. So we can collect the type of treatments and the response to treatment, how effective it was, and really study the genomic compositions of tuberculosis to understand the relationship between specific mutations in the genome and the effect on the treatment. We will also look at Ebola, where we will study a collection of all the Ebola samples from the 2014-2016 Ebola outbreak in Africa. And we will talk about how the different positions on the genome, once they mutate, as a response to this evolution of viral adaptation to new populations, right? If you think about viruses, what is unique about them is that they have a short genome, and to overcome any kind of host response, if we talk about the immune response, if we talk about the challenges of barriers between different organs, then to adapt to the responses and to the defense systems the host organism has, the virus has to mutate different regions of its genome. And as I mentioned, these types of viruses really evolve as communities. And so we can look at the community evolution and understand fitness of different mutations to study how viral adaptation can be correlated with, for example, geographic distribution or with outcomes, how lethal it is. Another aspect of these types of studies, the translational value of these studies beyond mechanistic understanding of how these viruses adapt and overcome these different defense mechanisms, is the ability to design vaccines. And so we know that this outbreak, the 2014-2016 outbreak, which was one of the deadliest outbreaks of Ebola, but today we know there's another outbreak of Ebola, it did lead to development of new vaccines. And the development of new vaccines is actually very critical for prevention of further outbreaks. And one of the limitations of the vaccines is this process of mutation. So if we train the organism immune response on a specific, to recognize specific patterns of proteins associated with the capsid of the virus, but the virus mutates and then the capsid doesn't look like that anymore, how do we actually address that problem, right? And the problem could be addressed by finding the most conserved regions and the ones that are critical for the viral replication cycle. So we'll talk a little bit about some of these concepts in the context of this Ebola project. Another study is an example of how we can use genome-wide association studies, typically studies that are performed on larger species, but in the context of bacterial genomes. And a good example is rhizobacteria, bacteria that forms on the roots of specific plants and can have an impact on how these plants can utilize the resources in the soil around them, transforming them to functional material for its growth and fruit production and others. So really a variety of projects ranging from biomedical to, you can say, behavioral, right, like the gut brain axis project, and to agriculture. So really we can see the impact of the study of microbiology and the study of metagenomics is very vast. Now, we'll also talk about how to take data from the public domain. How can one that, all of you joining the program, how can you find interesting data sets and understand what type of sequencing, how the data was generated, what is the context of the phenotypes that we are studying in the data? How do we translate that into a logical scheme of steps, just like we saw in this pipeline right here, right? All of these sections are represented by very logical step-by-step sections that you can utilize to address a very specific problem. So problems of really understanding the processing of data. How do we prepare data for analysis? And to prepare data for analysis, as we saw in this example, we have to understand what the challenges are, right? So by going through some of these examples, we'll see what the challenges are. We will also then try to understand the analysis part. This analysis part with phyloseq really allows us to study composition and compare composition between samples to then interpret how that might affect, for example, a certain condition. And that's a correlation that you need to identify as well. So we will learn about the data inputs, we will talk about the logic of the analysis, and we will talk about the data sets. How do we find those data sets? How do we go on repositories like the NCBI and like some of these other repositories which have quite a collection of samples to study the whole project. So just a quick overview of some of these materials. A lot of the materials that we will be going through in these online sessions are going to be focused on supplement information to the online courses that you can find here. Now, the courses that we have here start all the way from free courses that you can take. Anybody can join and take these courses. They are self-guided. You can see some of the topics that have been covered in these courses. And for example, this course is focused on this American gut metagenomic sequencing project. But you also get a lot more practice and a lot more guided program by joining the program that we have online, such as this program. So it's very important to understand the relationship between some of these different elements that I'm showing. And the relationship is that really, if you think of a program that you have access to somebody to talk to, ask questions, get feedback on your project idea, really understand what is it that you want to do and help you get there, that is the program. If you already know what you're doing and you would like to just understand how to apply a specific analysis method to a data set that you have, really what you can get is a research access to this platform where you can upload your own data, run some of these analyses, get the output, and you will still get feedback from our team on how to do it better and how to leverage some of the other resources that might take a while to learn about on this whole platform. And the other one is really, if you don't feel like you're ready for a program like this, you don't think that you will be working on your own project, you don't think that you need to understand the depths of the metagenomics sequencing and project examples, you would just like to explore what is metagenomics and how can it be relevant, you can start with a free course. For example, one of the courses that you can start with for free is our course called Introduction to Metagenomics. Introduction to Metagenomics right here. And in this course, we actually cover a lot of the basics in a very slow pace that you can adapt to your own schedule. If you have only a little bit of time, you can go through, and here you can see some of the introductory materials that you can benefit from. Now, in a second, I'll be wrapping up, and I would be happy to address any questions that you might have about the program itself, or talk about how some of the previous programs have helped some of the participants that we've worked with to achieve their goals. So you can learn a little bit about that. We've recently published a couple of blogs our team put out on how other participants, for example, you can see here Dr. Melissa Bratton from Xavier University in New Orleans, Dr. Zapico from Florida International University. You have some other people that have joined some of our previous programs and have really benefited from the research aspects, from the educational aspects, and really from understanding the logic of the analysis. So with that, I am going to wrap up the presentation part of the story and open up the platform for anyone, first of all, from our team that would like to add to what I've covered in this discussion. And feel free to talk about some of the more technical aspects of the program, including any scheduling questions about timing for international students or US-based students, as well as some of the aspects of how the program certificates work. You will get a certificate of participation based on your activity, based on what you do throughout the program and how many sessions you join. If you have any questions about the research platform and how that works, we can also talk about that. But also, of course, all of you are welcome to continue and stay in touch with us by email, or there is a chat box on the website itself where you can just type in your messages. So we'll be happy to answer any of those questions. So I'll pause now and let anybody else add to what I've said, or if you have any questions, please feel free to type in your question in the chat box. Mohit, would you like to add anything about the scheduling of the program?
Yeah, thank you, Ilya. First of all, I would like to thank everyone for joining this meeting. And as Ilya mentioned, that we'll be started this program today, and there will be another webinar coming up, and henceforth we'll have a lot of more webinars planned. So if I can share my screen and show you the guide you through the process. So we have an organization page with every program. So it's like a classroom where all the participants can see their progress. We'll be describing those in the next session as well. So I will just take you to the organization page and show you the schedule of the program. So here is the chat box that Ilya was talking about, where you can type in your questions regarding any queries that you have. So you can just click here and write about the question and ask us. So as for the next session, we will be starting on, we'll be talking on 16S metagenomic sequencing on 1st of November. And on 5th of November, we'll be talking about microbiome. And hence, again, on 8th of November, which is Tuesday, we'll be again talking about the analysis challenges, where we will be talking about short reads, HVR, O2Us, and we'll be discussing in depth the pipelines used for the analysis of 16S metagenomic data. And then on Friday, we'll be discussing mapping on databases. And on 15th, we'll be doing the data to pipeline that Ilya just showed you. So on 15th, we'll be taking a break where we will be letting you do a project. So those projects, as mentioned, there are multiple projects in our platform, which is, if you can see here, you can go into this and see the projects. So you'll be doing metagenomic project for a month. So we will be resuming on 10th of December again. So that the second phase will be more on the research topics. So we'll be covering bacterial population, GWAS, analysis of genome-wide association studies. And then again, on 13th, we'll be talking about viral genome detection in standard RNA sequence. So after that, we'll have few more sessions on phylogenetic analysis and drug resistance on 20th, and we'll be concluding on 23rd with a program with one-on-one discussion. So that would be all for this overview. So I'll give the stage back to the Ilya, or if anyone has any question, please feel free to ask us. So just stop the sharing.
Yep. Thank you, Mohit. Yes, anyone that would like to benefit from all of the aspects of the program, again, you're welcome to still register. We have several sessions planned, and all of these sessions are going to be recorded and shared with all of the participants that have registered. All of you will get access to the free courses, the paid projects, and also an educational license to the server platform that I just demonstrated, and a lot of guidance and one-on-one interaction with each one of our team. We have an experienced team that has been working in metagenomic data analysis for a while, and has participated in a number of publications, as well as these courses are being taught in a number of different organizations around the world, where we have college level and high school level programs that are also designed for faculty to learn about some of these topics and develop projects with students together with them. So again, feel free to ask any questions. If we don't have any questions, we will be wrapping up this session here in a few minutes. In about 5 minutes. And then after the session, we will be happy to share with you some of the recordings that we have for this session.
Hello.
So yes, hello.
I just wanted to know if I am registered for another program, it's the data analysis and the biomedical sciences. Will I have to register again for this course?
Yeah, you since you're already registered in another program, you can ask for a discount. We'd be happy to provide that for you. But this is a separate program that will cover completely different topics and will span 2 months. So just in general, to everyone, how these programs work, we have 6 to 8 programs per year, focused on different aspects of bioinformatics, starting from genomics, transcriptomics, metagenomics, epigenomics, machine learning for biomedical data, and an introductory program. So we hold these programs throughout the year. They're all several months long, from 1 month to 3 months. And so the payment that we are requiring for the program is actually based on 1 month access to all of these resources and the program. There is, however, an unlimited subscription that gives you access to all of the programs and the server platform to analyze your own data. So you can also inquire about that if you're interested in kind of pursuing multiple programs at the same time.
Okay, thank you.
Okay, well, thank you everyone for joining. Again, if you have any questions, feel free to reach out to us. I believe that she also posted a link to join the WhatsApp group. If you're comfortable using that for communication, we'd be happy to see you there. I'm very excited about this program to start. We'll be meeting again soon, and we are all excited to see you there. So hopefully you can email us or continue communicating with us in any way that's best for you, and we'll see you soon. And with that, I will end this presentation. And like we mentioned, we will send out the recording of this session and any other information that we mentioned during the session by email, and looking forward to seeing you again soon.